Okay, so first of all, start with introducing us. I'm Sam. I'm one of the immunology sales specialists here at ProImmune, with a focus on the Ankyrons. I've got a background in immunology, so I did my master's at UCL in immunotherapy, looking at rheumatic disease, and I'm joined by my colleague Louise, who is one of our application specialists and postdocs. I'll let Louise introduce herself to you.
Hi everyone. Hope you can hear me okay. Yeah, so I'm Louise, and I work on Anchoron applications and finding out really exactly what we can do with these. And my background is well, originally I was a biochemist, and then I did my PhD in immunology here in Oxford. And so yeah, now moved to ProImmune and having lots of fun with the Anchyrons, and hoping that we can convince you to get into them too.
Great, thank you, Louise. Good, so we'll get cracking. You have to forgive me if I'm a little bit nasal. I've got a little bit of a cold at the moment, so hopefully I'm not too bad. So to kick off with, I'll introduce ProImmune. I'm sure some of you are familiar with us already, but for those who are not, we're based in Oxford in the UK, and we've got locations across the world, so in the US and Singapore as well. We're an ISO regulated company, so we have a good quality management system and customers across the world. So we've got some academic partners and institutions that we've worked with that you'll probably recognize, and these are just the ones that we have publicly disclosed success with. Originally, ProImmune was founded by our founders to make MHC multimers for detecting antigen-specific T cells. And from there, the company grew into doing a range of different services for detecting immunogenicity of a range of different things, including therapeutics. And now, obviously, we're moving into the world of target binding reagents with the Ankyrons, which is something that we're rapidly growing in and very excited about, and obviously what we're here to talk about today. But it's sort of within the context of a range of other different products and services that we provide as well. So I think a good place to start with discussing the Ankyrons is to talk about some of the current challenges with research antibodies. So I'm sure many of you are aware there may not, if you are working on a particular research area or protein of interest, there might not be research antibodies available against the targets you're interested in. And if they are, they might be of poor quality or not very easily accessible. And if you choose to go down the route of custom antibody generation against a new target, this can take a long time, be prohibitively expensive, and also doesn't necessarily generate an antibody that works well in your particular application. It also will involve inevitably the use of animals if you're going down the standard antibody generation route, which leads to an antibody with an FC, an isotype background, and that's non-recombinant by default. So the nature of antibody generation at the moment is relatively pedestrian, and it can take quite a long time to generate antibody against a single new target. And that's really where the Ankyrons fit in. So the Ankyron is a target binding reagent based on an ankyrin-repeat scaffold. And an ankyrin-repeat scaffold is essentially a protein structure that mediates protein-protein interactions in nature. And we're able to use the ankyrin-repeat in our target binder to create a reagent that's much smaller than an antibody. So it's 15 kilodaltons compared to an antibody's 150, and we're generating it in a completely different way. So rather than animal immunization, we essentially have a genetic library of over 1 trillion different Ankyron specificities, and we can select from that library using ribosome display to identify clones that are medium to high affinity against a particular protein target of interest. And because we're generating them through this method, they're defined recombinant monoclonal sequences by default. And it's a significantly more cost-effective and high throughput generation process. So unlike a custom antibody, we can generate Ankyrons against many different proteins all in parallel, which is something that's very exciting for us. However, all the applications for the Ankyrons are exactly the same as a research antibody and the applications that you're familiar with. So flow cytometry, immunofluorescence ELISA, and Louise is going to talk you through some of the data that we've generated in-house and some of our collaborators have in a minute. So as I mentioned, we're selecting our clones using ribosome display. So and these clones that we identify have high affinity binding, even with a single site binding. And those are affinities in the picomolar to low nanomolar range. So on the right-hand side, you can see some data we generated in-house with some clones against PDL1, so an immune oncology target. And the Anchyrons against the PDL1 had affinities in the picomolar and low nanomolar range. And we can determine the precise affinity of our Ankyron clones using Cetylyzer, so solution equilibrium titration assays. And obviously, as I mentioned as well, the Anchyrons are not produced by immunizing an animal, which means that you're not constrained by immunological tolerance. You don't have to worry about any trying to raise an Ankyron against a self-antigen struggling because of tolerance, and there's no isotype or FC background to consider as well, which is an additional advantage of this recombinant system. And because of this generation platform and all of these advantages, we're able to investigate targets from across the tree of life. So these Anchyrons are of particular interest to people in the animal health sector as well, because at the moment there's sort of a dearth of reagents, and we're able to generate Anchyrons against a range of different target proteins from different animal species that wouldn't be commercially feasible to raise an antibody against. We are also able to do a guided selection process to distinguish between closely related targets. So with the ribosome display, we obviously identify binders to a particular whole protein or domains of a protein. But if you have two targets that have very high homology, you want to distinguish between them, you can use a guided selection approach. So a good example of this is raising Anchyrons against peptide MHC complexes or idiotype-specific Ankyrons. So in this case, you would screen a whole peptide MHC complex that you want to bind Anchyrons against, and then you would use a relevant MHC peptide complex as a negative selector to be able to identify Anchyrons that just bind to that specific MHC peptide complex. And on the bottom is some data generated by one of our external collaborators, Antonio Bertoletti, who we produced an Ankyron against an MHC peptide complex for. And on the far right, you can see in green is the staining of his MHC peptide positive cells after they've been pulsed with interferon gamma. The Ankyron format is also a very flexible format. So our standard Ankyron format has a V5 and a his tag. So a viral epitope tag is a V5. And you would use a secondary reagent, so an anti-V5 reagent, in order to stain the Ankyron in flow cytometry or immunofluorescence. But we can also produce those Ankyrons directly labeled with biotin flag tag, or with a range of different fluorophores, so our alpha lux range, which are equivalent to the Alexafluor range spectrally, and also HRP as well if you're doing immunohistochemistry or ELISAs. So it's a very flexible format, and we'll also talk a little bit more about that a bit later. So now I'm going to pass you over to Louise to run through some of the Ankyron application data and give you a feel for how we've been using the Anchyrons in-house, and how, as well as our collaborators, and some of their advantages in those applications.
Great, thank you very much, Sam. Right, so one of the things that we are doing is that we are building an Ankyron, ProImmune Ankyron cell atlas. And so by this, what we are doing is developing Ankyrons against ubiquitously expressed proteins throughout the cell. And so that way, if you're interested in where your favorite protein is in the cell, then you can use Ankyrons to find out which particular subcellular organelle is localizing with, to get an idea of perhaps what your favorite protein is doing within the cell. So all of the microscopy images I'm going to be showing you have been the cells which have been stained with the V5 tagged Ankyron, and then coming in with the anti-V5 APC conjugate. So the Ankyrons will be stained and shown up in red, and we also counterstain the nuclei with DAPI, so that appears in blue. And we take all of our images using a wide field microscope. So if, as part of the ProImmune Ankyron cell atlas, if you're interested to see whether your favorite protein is on the cell surface, then you can use our Anchyrons against CD44, which is a fairly ubiquitously expressed plasma membrane receptor. And we also have Anchyrons against junctional molecules such as the adherens junction proteins like Enkyrin and Ekyrin, and also tight junctional molecules such as CD31. We have Anchyrons to detect the endosomes and lysosomes, so Anchyrons specific to clathrin heavy chain, which coats the endocytosed membranes, and also the vesicles which move between the Golgi and the endosomes, and also Ankyron against some clones against MAP kinase 1 ERK, which is found in endosomes. And also, if you're interested in looking at lysosomes, then Ankyron against CD63 is proving highly specific. And if you look in our catalogue, you'll also see that there are Anchyrons specific for to various other endosomal and lysosomal markers such as LAMP1, CD9, CD68, and HER2. And we are currently developing a range of Anchyrons against the various stages of endosome and endosome maturation. So keep an eye out for those when they appear in our catalogue. We have Anchyrons which are specific for the ubiquitously expressed endoplasmic reticulum proteins, calreticulum and calnexin. These are chaperone proteins which will show up in every cell possible, and so serve as good markers for the ER in that case. And also, whilst we're on the subject of de novo synthesis organelles, we have Anchyrons specific to Golgi apparatus proteins. And shown here, we have RCAS1, which is found in the Golgi. If you want to look into the nucleus, then we have Anchyrons against various nuclear proteins too. And so here we can see nucleosomes using Ankyron, which is specific for the histone protein, and you can see the histone signal co-localizing with DAPI in the nucleus and appearing in the pink stain when it's co-localized. We've also, I should say, got some Anchyrons in development against other subcellular structures, particularly the cytoskeleton, which we'll be very excited to be using in the future. So to identify microtubules and also the actin cytoskeleton. So one of the things that we are finding, another great advantage of using Anchyrons, is that if you're carrying out immunofluorescence staining with Anchyrons, you find that it'll be much faster to carry out than if you're staining with antibodies. And so typically you'll save over an hour each time. And the reason for this is that if you're staining with antibodies, you have to typically block any non-specific binding, and then you have to wash quite extensively, usually for 5 minutes or so in between between incubations, because otherwise you end up with so much background that you don't get very good pretty pictures. However, we find that with the Ankyron staining, we just don't need to block beforehand. We just include a blocking reagent like serum in with the Ankyron and the secondary, and also we don't need to wash so much, and we think that's simply because the Anchyrons bind their targets with such high affinity, and also they're that much smaller, so you don't need to carry out so many wash steps. So it makes life a lot easier and a lot faster as well. One of the other things, other applications that we are investigating is using Anchyrons to look at cell activation. And so an example of this is using Anchyrons against the adhesion molecule VCAM1. And this is absent from resting blood endothelia and as we show here, human umbilical vein endothelial cells. But when the cells are stimulated with TNF-alpha, you can see a massive increase in VCAM1 expression, shown here in red by detection with using the anti-V5 APC. And we can show this by immunofluorescence and by microscopy, but also the Anchyrons work very nicely in flow cytometry as well, as you can see in the panel here on the right. we are actually, I should also point out that we're looking at developing other activation markers, and we already have Anchyrons against the T cell activation marker CD69, and also dendritic cell markers which come upon that activation CD80 and 86. So as I showed you with the VCAM slide, Anchyrons can be used very effectively to label proteins by flow cytometry, and they're proving very popular with some of our collaborators because they work on labeling proteins against which there's no reliable antibody at present. So for example, the transcription factor HIF1 alpha, and currently there are no reliable antibodies that work very well for flow cytometry. And so our collaborator, David Withers in Birmingham, he was very happy to use our Ankyrons to look at HIF1 alpha expression in mice. So what he did was he took some C57 black mice and injected them with colon carcinoma cells into the flank, and then waited for the tumors to grow. And then harvested the tumours and also the spleens and isolated NK cells, and then stained these for HIF1 alpha using this Ankyron clone. And he found that the HIF1 alpha expression he could detect it in splenic NK cells, but there was a massive increase in the tumor NK, and so this is statistically significant when you look at the bar charts on the right-hand side. So the take-home message from his experiments was that there was low HIF1 alpha expression in NK cells in the spleen, but it increased upon in the tumor NK cells, and the Ankyron worked very effectively. So some of our other collaborators have been using Ankyrons to identify spike proteins against SARS-CoV-2. And so we sent them a selection of Ankyron clones, some which recognize the original form of, so the Wuhan form of SARS-CoV-2, and then the various variants, so Delta and Omicron. And so they were using this biolayer interferometry, which is an optical technique for measuring macromolecular interactions by looking at the interference patterns of white light on the surface of the biosensor tip. So what you do is you immobilize one of your molecules that you're analyzing on the sensor tip, and then you put on your second molecule in solution, and then see what binds effectively. And so they were able to show that the Ankyron clones could bind very specifically to the individual spike proteins, and there was not cross-reactivity between the spike proteins. For example, this particular Ankyron clone would only bind the wild-type Wuhan version of the spike protein, and did not bind MERS, the Middle East respiratory syndrome virus instead. And also they used BLI to determine the affinity, and as you can see, the KD value of this is 90 picomolar, so it is incredibly high affinity. They also were looking at whether the Ankyron clones could neutralize SARS-CoV-2, and so what they were doing this, they were using a pseudovirus stocks, and then looking at the titers after incubation with these various Ankyron clones. And as you can see, some of the clones particularly demonstrated very effective neutralization through binding to the spike protein. So this, this is pretty exciting. It raises the possibility that Anchyrons, a bit like antibodies, really can have antagonistic or agonistic effects. So I think this would be a nice thing for us to explore, and also our collaborators to look at these effects in the future. Then the final bit of data that I'm going to show you today is that's been kindly provided by our collaborators in Bern in Switzerland, and they are interested in a is a protein called GABARAP, which is a ubiquitin-like modifier, and it's essential for later stages of autophagosome maturation. And they are particularly interested in this pathogen called Plasmodium berghei, which infects rodents. It's a rodent malaria parasite. And their problem was that there were just no antibodies against GABARAP, so it made studying GABARAP very, very difficult. However, they used so we developed some Ankyrons for them, and they've been using this very successfully. And as you can see here, first in the top panels, these are HeLa cells, wild-type HeLa cells, which have been infected with the Plasmodium berghei and then imaged 24 hours later. The parasite is known to reside in the PVM membrane, shown in magenta. And as you can see here, the Ankyron, which binds GABARAP, lights up in green and co-localizes very nicely with the PVM. And you can also see the parasite, which is a much smaller little blob of DAPI in its nucleus, right next to the host cell. And what was also great was that when they knocked out GABARAP from HeLa cells, we could still see the magenta staining of the PVM, but it loses the colocalization of GABARAP. So we can show that, well, they could show that the binding is specific for GABARAP. So they were able to show, first of all, that GABARAP co-localizes with the PVM, but also this makes us and them think that one of the great things to take the Ankyrons in one of the directions we could take in the future was to use Ankyrons to track intracellular events. So with that, I'm going to leave you back with Sam, and he can talk to you a bit more about the future directions and applications. Thank you.
Yes, thank you, Louise. I hope that's been a good introduction to the sort of data that we and our collaborators will be generating with the Ankyrons, and giving you a feel for how they're being used at the moment. To go through some future directions, as I mentioned earlier, the Ankyron format is very flexible. So aside from the direct labeling, one of the other options that is available going forward is to link multiple Ankyron binders together, so for multivalency to create bispecifics or daisy chained Ankyrons. Obviously, as single site binders, the Ankyron still have high affinity, but there may be applications where you want to increase the avidity of interaction between the Ankyrons and your target of interest, or you want to do receptor clustering or something along those lines. Then that's absolutely something that the Ankyrons can do. And I think also, particularly the bispecifics, it's something that's going to be very exciting going forward. The ability to bring a reagent like that to the research space, because currently at the moment generating a bispecific is something that you would primarily be doing if you're looking to generate a therapeutic, not so much something you would be generating as a research tool. So that's something that's very exciting, and also some new applications as well that we're looking towards. So intracellular live cell imaging, super resolution imaging. The Ankyrons are very small, as we mentioned, so the binding site is close to the fluorophore, so you get good resolution of imaging. So they'd be great for an application like that. Ankyron multiplexing, and of course, rapid catalog expansion, increasing the number of specificities that we have to offer. So we already have about 1,500 different proteins which we've raised Ankyrons against, and that represents about 7,500 different clones. So we have multiple clones available against the different targets of interest, and these are all visible on our online catalog. And as I mentioned, our standard format is the Ankyrons are produced with a V5 and a his tag. On our catalog, you can see all the clones were identified as specific target binders to a particular protein of interest. For some of our clones, we have additional validation data that's generated in-house or by a collaborator, and these we term our qualified clones. And that data is visible to view on our website on the right-hand side under the applications, and you can see which particular application the clone has been validated in. So, for example, this VCAM1 clone has both immunofluorescence and flow cytometry data. Aside from the catalog Ankyrons that we have available to offer, we also have a custom workflow as well. So we can generate Anchyrons against new targets of interest using ribosome display in exactly the same process. There's two ways in which we can go about this. Obviously, we need the target protein of interest to generate the Ankyrons, and either a customer can provide the target protein to us. We relatively early on discovered that quite a lot of people don't necessarily have the purified version of the target protein that they're interested in. So we've also developed a process in-house, a platform, Ankyron X we call it, for synthesizing target proteins of interest. We have both prokaryotic and eukaryotic cell lines we can use to express the target proteins. And all we need to do this is either a UniProt ID or a sequencer, like a FASTA file. And we envisage this workflow fitting in very nicely with the rise of multi-omic, transcriptomic approaches to research. So, for instance, you might identify multiple proteins of interest that you find upregulated in your particular research area, and then you want to do functional studies and or identify if they're identify the function of those proteins, and then all we need to do that is UniProt ID, and then we can synthesize those proteins, generate Ankyrons, and provide you with target binders for your research. So typically out of the custom generation, we'll identify many clones, but in the first instance, we'll send a customer up to 6 to test in their application and identify the ones that work best for them in whatever application they're using. So much like an antibody, some Ankyron clones might work slightly better in one application over another, but that's why we'd send multiple clones for somebody to test and identify the ones that are working best. Those will, in the first instance, be with a V5 and a his tag, and then once the clones are identified that are working well, we can move to doing the direct labeling and direct conjugation. So just to sort of summarize, the applications of the Ankyrons are all the same as a research antibody, so immunofluorescence, flow cytometry, ELISA, immunoprecipitation, cell culture, and more. And using this platform of ribosome display to identify high affinity target binders, we're able to generate reagents at high throughput and in a very cost-effective manner, and also able to generate Ankyrons against challenging targets such as peptide MHC complexes. And this is enabling the interrogation of diseases and species areas that have not previously been possible with research antibodies in the same way. So just to finish off, I'll do a little bit of a comparison between the Ankyrons and the antibodies. As I mentioned, they're much smaller, and Louise has gone through some of the ways in which that brings about advantages in your research applications. They're obviously selected from a Terra library rather than animal immunization. This also fits in nicely with the move towards or the move away from using animal-derived reagents in research. So it's good for your grant application. And they're available as a range of different custom conjugates. So hopefully that's been a good introduction to the Ankyron technology and given you a feel for how you might be able to apply them in your research. I'd be very happy now to answer any questions you might have had throughout the presentation or about the Ankyrons in general. Can I use Ankyrons for cell separation like Ankyron conjugated magnetic beads?
Oh, well, actually, it's interesting to say that because that's something that we are looking into right now, actually, because we are keen to use Ankyrons to pull out monocytes. So we've got some good Ankyrons that bind CD14 and also CSF1R, which work very nicely for flow cytometry, and we want to check that they work well as we expect them to for magnetic bead selection. So that's something that, yeah, we're definitely keen to do, and the idea I think would be that if we could optimize those conditions and maybe even get the direct conjugate of the clone with the magnetic bead, then that would make the life a lot easier for the customer. In the chat, is it possible to obtain the nucleotide sequence of a specific Ankyron? Unfortunately, at the moment, the sequence is something that's our intellectual property, so we wouldn't be able to disclose the sequence for expression. But I mean, depending on what application
it is that you're looking to do, we could look at ways in which we might be able to help you achieve those research goals without necessarily providing the sequence to you. So if you have any specific questions about your application, we'd be more than happy to discuss them with you, and how we might be able to help. And it's a question about whether which targets make best Ankyron targets, and whether there's any difficulties synthesizing Anchyrons against particular targets. So there are two separate things to address here. So first of all is targets that might be challenging to synthesize on the Ankyron expression platform. So if a customer is not providing a target protein to us, then we can GPCRs or multipass transmembrane proteins can be sometimes difficult to express on our Ankyron expression platform, and therefore at the moment that's not one of our top priorities. But it's not because you wouldn't be able to raise an Ankyron against it. It's actually the expression of the target protein that's challenging for us. So that's one thing to consider. In terms of actual Ankyron targets, the Ankyrons are binding conformational epitope. So anything that's got a good stable structure will form a good Ankyron target. Anything that's very small or is a floppy peptide or unstructured domain might not be the best target to raise an Ankyron against. But generally things with a stable structure will form a good epitope for Ankyron binding.
Actually, I should say that we put our protocols on our website. So if you do buy some Anchyrons and have a play with them, then our protocols are probably quite a good starting point, really. And also, if you are having issues, then get in touch, and we'll try our best to troubleshoot.
Unless anybody else has any further questions, I think we can wrap it up there. Well, thank you everybody for coming. Hopefully, as I say, it's been a good introduction, and thank you all for your interaction and your questions. And we've had some good questions, and hopefully you'll be able to think of some good ways to apply the Anchyrons in your particular application.